The magnetic breather pump, or MBP, is a device specifically for the treatment of malignant gliomas and other aggressive brain tumors. Its goal to achieve improved patient response using microfluidics to increase diffusion and penetration of medications to the target site. The pump consists of a small, round, flexible 2-layer polymer pouch filled with a medicating solution. An array of hollow spines radiate from the inner membrane through the matching pores on the outer skin. The pump cycle is created by a solenoid composed of a neodymium magnet and electromagnetic winding fastened to the head and base of the pouch respectively, forming an electrostatic muscle as they attract and repel. A pleat on the inner membrane at the base of each spine retains a shape memory which permits a telescoping action, causing rapid extension and retraction of the spines during the pump cycle. As the two components contract, the volume in the pouch is compressed, forcing medication through the spines on the supply stroke. On the intake stroke, the spines retract, and the expansion of the pouch creates a suction of the surrounding cerebrospinal fluid, which can be analyzed to assess tumor condition. The fluids and medication pass along a flexible tubing joining the pump head valve to the connector seal, which braces the pump in place on the skull. The supply and sample return outlets attach to lines running through a catheter connecting the pump pouch to the external pump control device. The external pump control can deliver up to 4 ampules of different medications in combination according to the specific needs of the patient. Independently controlled piezoelectric pumps extract the drugs through needles which penetrate the ampules. A Bluetooth transceiver enables a physician to remotely access the memory to retrieve data and alter the drug regimen if desired. A lithium battery powers the device and is recharged via wireless induction. Cerebral fluid drawn into the pump is sent to a lab-on-chip which monitors levels of VEGF, or vascular endothelial growth factor, for indications of progress or regression of the tumor burden. The cerebral fluid is infused with monoclonal antibodies which specifically bond to the VEGF protein. Patient progress can be measured by spectroscopic impedance analysis of the sample medium to detect the occurrences of the resulting molecular chain. Fitting over the main board is the induction coil section which allows wireless recharging of the device. The malignant glioma is the most common type of primary tumor of the central nervous system. For this type of tumor there is currently no cure, despite improvements in surgery, chemotherapy, and radiation therapy. At the most advanced stage, a glioblastoma multiforme spells a life expectancy of 12 to 15 months. As the tumor grows, it produces vascular endothelial growth factor to stimulate an increased blood supply. After the cranium of the patient has been opened and the skull and dura have been successfully breached, the tumor, or as much of the tumor as possible, is removed. The soft cranium pump is then placed in the resulting cavity, and the skull cap is reattached. The pump is positioned on the patient's brain beneath both the dura and skull. The delivery catheter leads from the connector seal away from the pump down the back of the neck